Hi, this is Dr. Claire, and today we're going to be talking about Mendelian genetics. First, we need to talk a little bit about genetic terminology so that we're all on the first page, and then we'll talk about some patterns of Mendelian inheritance. All right, so first, let's talk about how the genetic code is found within most organisms that we'll be studying this semester. So mostly we're going to be looking at eukaryotic organisms. And eukaryotic organisms have their DNA arranged into linear chromosomes. So these long strands of DNA that are all wound up in the nucleus, and they are in these chromosomes. So the chromosome number can actually vary quite a bit depending on the species. So we as humans have 46 different chromosomes, so we have 23 different types of chromosome, and we have 2 of each type. Okay. Other species have much fewer. Fruit flies only have 8. The kingfisher, which is a bird, has 134 chromosomes, and even the lowly potato has more chromosomes than we do at 48. So the number of chromosomes that a particular species has may or may not be associated with its perceived complexity. Okay. Now, if you're looking at these organisms, another way you can describe them is by describing their ploidy level. And ploidy basically refers to the number of copies of each type of chromosome that they have. Most organisms we're going to be dealing with are diploid. That means that they have 2 copies of each chromosome. Okay? And so, for example, we as humans have 23 different types of chromosomes. We have 2 copies of each, hence 46 total chromosomes. Okay? Some rare organisms are actually haploid, meaning they only have 1 copy of each chromosome. However, more commonly, the haploid state occurs in the gametes. So sperm and eggs are haploid. So when a diploid organism, such as ourselves, produces either sperm or eggs, we have to half the number of chromosomes that we have, and then we only put half into each sperm and each egg, okay? And we put 1 copy of each type of chromosome into those sperm and eggs. Okay, so let's take a closer look at these genes. So a gene is basically a chunk of DNA in the genome, and that gene has information in it about some sort of characteristic. Now, genes can be identified by 2 different things, both their location on a chromosome, so that they're always found at a particular spot on a particular chromosome, so they're always in the same location within the genome. And then they can also be identified by the protein product that they produce. Most genes produce some sort of protein product. Okay. Now, genes, while they're always found in the same spot, they may not always be in the same form. So different forms of the same gene are known as alleles. And alleles can specify a particular form of that particular trait. So for example, you might have a gene for eye color, and different alleles could code for blue eyes or brown eyes. Okay? And the difference between those alleles is actually going to be in the DNA sequence, in the nucleotide sequence within the gene. So there's going to be a slight difference there. That's going to have an effect on the proteins that those genes code for, and that's what actually causes the difference in the trait. So if you're looking at 2 homologous chromosomes, each pair of homologous chromosomes will have the exact same genes in the exact same order, but they may or may not have the same alleles. So they might have different forms of those genes. So for example, if we look at this example down here, we have 2 homologous chromosomes. They have the same genes on them. They have a gene for eyelash length and a gene for hair color. And then this particular individual that we're looking at here has 2 different alleles for the hair color gene. It has 1 brown allele and 1 blonde allele. But it has 2 of the same alleles for the eyelash gene. So what you would say with that is that this individual is heterozygous for hair color, meaning it has 2 different alleles for the hair color gene. But this individual is homozygous for eyelash color, meaning it has 2 of the same alleles for the eyelash gene. Okay, so an individual can be heterozygous and homozygous at the same time. Okay, all right. Now, when we're describing the genetic composition of a particular individual, we usually refer to its genotype. And the genotype is the combination of alleles that it carries in its chromosome. So usually we arbitrarily or somewhat selectively pick some sort of letter to specify a gene with. it doesn't really matter what those letters are. If you are looking at a genetics problem and the letters that we use to symbolize the gene are not specifically specified, you can make up letters. As long as you define them, anything is perfectly acceptable. So, those letters can be things like A or B or C. And then the different alleles within the same gene are designated by slight changes in those letter designations. So quite frequently, if there's only 2 alleles for a particular gene, we'll use uppercase and lowercase. So there's a lot of uppercase and lowercase alleles. That's very common. If you have more than 2, uppercase and lowercase doesn't work very well, so you can use subscripts or superscripts. So here, A sub 1, A sub 2, A sub 3, or even C sub B, C sub O. All of these are acceptable ways to designate alleles within a particular gene. And then when you look at the genotype, anytime you have 2 alleles that are the same, you're looking at a homozygous genotype. Anytime you have 2 alleles that are different, you're looking at a heterozygous genotype. Okay? All right. Okay, so that's the genotype. Now the phenotype is the outward expression of that particular genotype. Okay? And sometimes it's pretty clear. So let's take the hair color gene. You have an allele for brown hair and you have an allele for blonde hair within the hair color gene. Now if you are, and here we've designated brown hair as big B and blonde hair as little b, okay? So if you are big B, all you have is brown hair genes, or alleles rather, all you have is brown hair alleles, so you're gonna have brown hair. If you have little b, all you have is blonde hair alleles, and so you're going to be blonde. Now, if you have big B, little b, it's a little more unclear because you don't know the relationship between those 2 alleles. And the phenotype is going to depend on that relationship. Now, a lot of these relationships are what we call dominant recessive relationships, where one of the 2 alleles will, if it's present in the genotype, will dominate and the other one will not be noticeable in the phenotype. Brown and blonde hair is generally one of these cases. So if you have a brown hair allele and a blonde hair allele, you'll usually have brown hair. But sometimes it's more complex than that. So you can have cases where you have, let's say in rabbits, there's an example in rabbits where there's a black allele and a white allele, and when you have one of each, you're gray. So it just depends on the particular gene that you're looking at, okay? All right, so let's look at an example. So let's say we have, we're looking at some peas, and the reason we use peas is because of Mendel. He came up with a lot of these Mendelian rules of inheritance. They're named after him. So we have these peas, and in these peas we're going to be looking at flower color to start, all right? So there's 2 alleles for flower color in these peas. There is a purple allele, we're going to call that big P, and there's a white allele, we're going to call that little p. So we got 2 parents here. We got the big P parent, P, and a little p parent. And so big P is purple, little p is white. Okay? If we breed those guys together, we breed the big P parent with the little p parent, we get a population of offspring. We call this the F1 generation. That stands for filial 1. Filius is the Latin word for son, so as in like your child. So the F1 generation, we'll all get 1 allele from mom and 1 allele from dad. Since mom only has big P and dad only has little p, every offspring in that cross is going to be big P, little p, right? The F2 generation, so if you take those big P, little p, those babies, and you let them grow up and you cross them with each other, then you get the F2 generation. Okay, and the F2 generation is going to have a mix. So there's going to be some that are big P, some that are big P, and some that are little p. Okay, so when we're looking at this set of traits, these are our genotypes here. So we have a mix of genotypes. These are our phenotypes. So what it actually looks like is the phenotype. We've got some homozygotes. We've got some heterozygotes, and you'll notice in this case all of the heterozygotes, so the ones that have 1 big P and 1 little p, their phenotype is purple. What that means is that the purple allele is dominant to the white allele. So the purple allele is dominant, the white allele is recessive. The heterozygote individuals are indistinguishable from the homozygote dominant. They're all purple. They all look exactly the same. And it's a very common pattern of inheritance, okay? All right, so So when a parent produces offspring, those alleles, the two alleles that the parent has, segregate, and the alleles divide, and each gamete gets one allele. Okay, so this is called the segregation of alleles. So whenever you have a gene and you've got two alleles for that gene and you're trying to figure out the gametes that that individual is going to make, you have to take those two alleles and you can make either type of gamete. Okay, so for example, if an individual is heterozygous, meaning that it has two different alleles for that particular gene, then 50% of its gametes, of its sperm or its eggs, are going to have the dominant gene, and 50% of its offspring or its gametes are going to have that recessive gene. Okay, so the offspring of heterozygote parents have equal opportunity to inherit either of those alleles. Okay, so far so good. Okay, all right. So so this is going to be important in terms of predicting what kinds of offsprings you can get from any particular cross of parents. And one way to make that prediction is to use what we call Punnett squares. Okay, Punnett squares are a very useful tool. So it's a good way to predict your genotypes and your phenotypes. So let's take a look at a Punnett square here. So let's go back to our first crossing that we made there with the big P parent and the little p parent. So the first thing you want to do is you want to make your Punnett square. So your Punnett square is going to have enough cells across and enough cells up and down to list every possible gamete that that parent could create. Okay, so in this case, we have one gene with two alleles. That means that the parents can only produce two possible types of gametes. All right, so if we're looking at the purple parent, in this case we're saying the purple parent is the male, so he's producing pollen or sperm. And if he's big P, what kind of sperm can he make? He can only make big P sperm because that's all he has. So he makes big P sperm. Same goes for the white parent. This is the female. She can only make little p eggs because that's all she has. All right. So you put the sperm along one side of the Punnett square, you put the eggs along the other side of the Punnett square, and then you combine them together into the squares. So you get your genotypes of the offspring within each square. Okay. So far so good? All right. Now in this cross, one parent is only producing big P, one parent is only producing little p, so 100% of the offspring are big P, little p, heterozygotes. They're all purple, and they all have the same genotype. Okay? So far, so good? All right, now let's look at the F2 generation. So now we're going to take those heterozygotes from the first cross, and we're going to breed them together. Okay? Now, it's a little bit more complicated. So we've got these two parents. They are the same as each other. So what kinds of sperm and eggs can they produce? So now we've got a heterozygote. So it can either produce big P sperm and big P eggs, or big P sperm and little p sperm, or big P eggs and little p eggs, right? Depending on whether they're male or female. All right, so here's our gametes. And then you fill in the boxes in the Punnett square. So there's our fill-in. So in this case, we wrote the egg first and then the sperm. So big P, big P, big P, little p, big P, little p, little p. Is there a difference between little p, big P, and big P, little p? No, they're exactly the same. So they're both heterozygotes. It doesn't matter which order you write them in. They've got one big P, they've got one little p. They're exactly the same. Okay? All right, so now we've got a diversity of genotypes among the offspring. We've got a quarter of the offspring are going to be big P, half the offspring are going to be big P, little p, and a quarter of the offspring are going to be little p. So we have this 1 to 2 to 1 ratio of genotypes. Now you remember what we said about dominance in this trait. Which color is dominant? Purple, right? So when you look at at the phenotypes of these offspring, what you're going to see is that 3/4 of them are going to be purple, and you're not going to be able to tell those heterozygotes from the homozygote dominance, and a quarter are going to be white. Okay, so you got a 3 to 1 ratio. All right, hopefully this sounds vaguely familiar. You've seen Mendelian Genetics at some point in time in the past, and this seems roughly familiar to you. Now, let's look at something a little bit more complicated. What happens if you have more than one gene? So you've got two different genes, and each of these genes has multiple alleles. So now we're going to take a couple of different scenarios, and follow them through a Punnett. Well, we're not going to do a Punnett square, but we're going to talk about what happens with these. So now we're going to look at two different traits. We've got seed color and we've got seed shape. So we have yellow or green, and we have round and wrinkled. Okay, so you can be being yellow and green is mutually exclusive. Remember our probability rules. You can't be yellow and green at the same time. But you can be yellow and wrinkled. So that the traits within a single gene are mutually exclusive. The traits between genes may or may not be independent. We'll talk about that in a minute. Or actually, we'll talk about that in class. All right, so if you did a monohybrid cross, like we did with the pea flowers, and you cross a yellow pea and a green pea, you get all heterozygotes and they're yellow. So what does that mean? It means yellow is dominant to green. And if you cross the shape peas, you cross round peas with wrinkled peas, you get round peas. So that means that round is dominant to wrinkled. All right? So these are both monohybrid crosses. What if we mix them together? So we take a yellow and round pea and we cross it with a green and wrinkled pea. What do you think is going to happen? You're going to get yellow round offspring that are heterozygous for both color and shape. Okay? That makes sense? So you got these heterozygous offspring, all of them are the same, and they get the big Y allele from mom, the little Y allele from dad, the big R allele from mom, and the little R allele from dad. So they're heterozygous for both traits. Okay? Now, here's the confusing thing. What happens when you cross two of these double heterozygotes together? What do you expect to get? And what turns out to happen is that you get a mix of the different traits. You get some that are yellow and round, you get some that are green and round, you get some that are yellow and wrinkled, you get some that are green and wrinkled, and they occur in a 9:3 to 3:1 ratio. Now we're going to explain how this works and why this works in class, and we'll do a Punnett square for this in class, and I hope to see you there on Monday.